All right, everyone. So welcome to section 2, where I'm going to be going over lectures 4 and 5. Let's just step right into it. Your guys' exam is on the 19th, so about a week from this recording. And yeah, I'll try to get up lecture 6 over the weekend and do like a slight review. We will have dedicated review sessions and also Q&As as well. So there will be a lot of times that you guys can just show up to the review session. You can show up to Q&As. You can show up to ad hoc office hours as well. There's going to be a lot of times for you that us TAs are going to provide. So be on the lookout. Okay, so going into lecture 4, we're going to go over protein function, and if you guys remember, lecture 4 was all about the ligand association, dissociation, hemoglobin, myoglobin, all that stuff. So we're going to take a deeper examination of it. So just going into the ligand binding intro. So just to keep it very simple, superficial terms, you're going to have a protein and you're going to have a ligand. And essentially, the protein is going to have its corresponding ligand, which it wants to favorably bind to it. So you're going to form this complex. It's called the PL, and when we talk about enzyme kinetics, we're going to talk about ES. E, enzyme, and then S for substrate. So just know that these do correlate. But essentially what I'm trying to bring into light here is that the association constant, essentially, it's in the name. It's the, what's it called? It's the equilibrium constant for the protein and ligand binding together to form this complex. So as you can see over here, these equilibrium constants, recall from Chem 330, that it's always the products divided by the reactants. And so in this instance, the association means that our products are going to be the associated complex, and then the reactants are going to be them separate. So that's what we see here. And then dissociation, it's the reverse in the name dissociate. So our reactants are technically going to be this PL rather than products. Does that make sense? So this is now our reactants. That's why we see it at the bottom. And then for the separated version, the protein and ligand separated, we see that as our products on the top. So just a little brief overview. You don't really need to know these constants. They're just interrelated, but let's move on. So in terms of ligand binding, so just to clarify really quick, theta represents the percent of occupied binding sites, which, to keep it in very simple terms, let's just say we had 100 proteins, right? And that would be our total binding sites, right? Let's just say we had 100 proteins. And let's say that 50 of those proteins were occupied, right? So we have 50 divided by 100, which means that it's half, or 0.5. That's where we see here. And I'm not jumping the gun. I'll explain everything. But this is just to keep it in simple terms. So if let's say we had 100 proteins again, but we don't have any substrate, therefore we have 0 occupied binding sites. That means our ligand concentration is 0. We don't expect any of these proteins to have the ligand on it. So we would expect theta to be 0 because the top would be 0, and this would be 100. Now let's say we add enough ligands that every single protein becomes occupied, and this is called saturation. And if all the proteins are saturated with their ligand, then it's 100 binding sites occupied out of 100 total proteins. So then theta becomes 1. So that's something to know, like just keep it conceptually in your mind, is when theta is 0, we have no binding sites occupied by the ligand. So it means that our concentration is either 0 or very minuscule, that it's basically essentially 0. And then when theta is equal to 1, that's when we think of, okay, all the total binding sites are occupied. So again, there are 2 equilibrium constants between the protein ligand and ligand plus protein that are separate, but they describe a similar concept. And that's the association constant, which we briefly went over before, and also the dissociation constant. And just for a quick little note, we don't typically talk about the Ka in biochem or enzyme kinetics. It does come up from time to time, but the one that we'll be focusing on is Kd, especially for the next lecture, lecture 5 of enzyme kinetics. So something to keep in mind is that we typically talk about this in terms of dissociation. The concentration at which, or the affinity of the PL constant to separate and dissociate into its starting reactants or products in that regard. Not to confuse it, to go in the reverse direction. But essentially, the Kd is going to be equal to the ligand concentration when half of all the binding sites are occupied. Theta is equal to 0.5. So if we were looking at this diagram, right, and well, let's just say it's 10, right? 10 is the max, and then 0 is the minimum, obviously 0 ligand concentration. And let's say this is about, I would say that's like 2. No, 1. Let's just say this is 1 and nanomolar. This is arbitrary units. 1 nanomolar of our ligand concentration. So what we find here is that our theta is half when we have this 1 nanomolar concentration of our ligand. So what that means is the dissociation constant, the concentration at which 50% of the binding sites are occupied, is going to be 1 nanomolar. I'll repeat that again. So when theta is 50%, that means we have half the binding sites occupied. And at 50%, we need to find out, okay, well, what is the Kd? And we know conceptually that the Kd is going to be equal to the ligand concentration at which theta is 0.5. And again, the concentration of ligand is equal to KD when theta is equal to 0.5. And this is because this graph is hyperbolic. It is not linear, meaning we can't just take the slope or whatever. We have to look at this 0.5. There's a whole derivation of it. You just need to know this. I can just have it memorized, essentially. And yeah, Ka and Kd are inverses of each other, much like Ka and Kb when we're talking about acid bases. Not to throw you in a different whole concept, but it's good to tie things together. And something to keep in note is that the higher the Kd, the lower the binding affinity for the protein and ligand. And what this means is that if, for example, we have two curves, right? We have one that it's this one, the black one, and then let's say we have another curve that's out over here. It's below it, but the Kd is, let's say, at 5, right? Well, this means that if we start from 0, right, and so we have the two Kds. We have one at 1, and then the other one is at 5. So we have one here at 1, which is 0.5, and let's say the other one is arbitrarily here at the 5. Well, if we're starting from 0, we have to add such little ligand for the first line right here that it's going to have higher affinity. Does that make sense? Whereas the second line that we're saying that the theta being at 0.5 is at 5 ligand, we have to add more. So essentially, it's like we're adding more of the ligand to compensate for the low binding affinity. We have to add more. Hopefully that makes sense. That's just another way of explaining it. But essentially, you can think of Kd as an inverse of binding affinity. So if we say that something has high Kd, the binding affinity is low. And inversely, if we say that something has a low Kd, the binding affinity is extremely high. So something to keep in mind. These are two different ways to think about it, as if it's a little bit confusing. Okay. So this is a quick practice on estimating Kd for this logarithm plot. So essentially, look for the way you would normally approach these questions is pay attention to your units. So like, for example, we have X, it's an arbitrary value. We would give you like the concentrations, all that stuff. So you would know the units and everything. But we're seeing here that's at 60, 120. This we're assuming is 0 because of the ticks and all that. So we'll assume that. And then it goes up to 500 and it caps out at 500. So this is the Vmax right here. And we'll go into this later, so it'll make sense. But the cap is at 500 because it's not increasing the slope anymore. So because it's at 500, what's half of that? 250. So we need to estimate approximately 250 from this line. And it looks like it's about here. I would say right here designate as 250. And then from here, we would just basically, if I was doing this on exam, draw a line across from 250. Whenever it hits the line, then I would draw straight down. And essentially, that's when you draw straight down from the halfway point when theta is half, or in this case 250, then we see that, we're at a concentration of about 15, let's just say. And so that's what our Kd would be, is 15 micromolar, nanomolar, whatever the units are. And yeah, that's just the process of how you would quickly estimate it. And yeah, interpreting binding curves, just to talk about this again. So again, y-axis, find the value half of the max. So you're going to have theta, right? You're going to get 1. And they may not even give you the theta. They may give it to you in the graph like this one previously, right? Where it's units instead. And it'll be like 500. And obviously that's more than 1. So then and when you find that there's this like cap or there's the stagnation at the very end of this curve, that's when you know you're at the maximum. And that you can assume is 500. That'll be the top portion. So you just take half of that. And that's how you would find this theta is equal to 0.5. Again, that's the whole point of theta, is that it's the fraction of total binding sites that are occupied. So we have the number of occupied binding sites and then the number of total. And it's a fraction essentially. And then the plotted line is move right to find the point of plot that corresponds to theta. So you just take the theta half, you move to right. Once it intersects the plot, then you just go straight down, and then you'll find your Kd correspondingly. And again, Kd is the concentration of ligand that allows for the protein to be half saturated, meaning half of the sites are occupied. And again, leftward shifts of the plot. So if this line was to move a little bit like this, like essentially the Kd, you would reach the theta is equal to 0.5 at a lower concentration of ligand. That's increased binding affinity. This is because, again, you need less of the ligand in order to reach half saturation, which means that the ligand likes to bind onto the enzyme. You need less of it. Whereas if you have a right shift plot here, if this, again, using the previous example, if we were at 5 molar we needed 5 micromolar of this ligand, right? Instead of like 1 or half or whatever. If we needed more of it, that means it binds much more loosely. We need more of the ligand in order to get to half of the occupied binding sites. So something to keep in mind. Right here, pay attention to the x-axis. This is just, again, what I was stating before. just keep in the rule that it's like inversely related, inversely proportional. So if the KD goes down, your binding affinity goes up. If your KD goes up, your binding affinity is going to go down. So they're inversely proportional. So if you ever get confused or whatever, just know that they're inverses to each other. And yeah, and this may seem counterintuitive, but we can eventually associate moving left on the graph with decreasing and right with increasing. But be precise about what is increasing, decreasing when the light shifts and how that relates to other nomenclature, other concepts, right? So KD does move left, it does decrease, but that means that the binding affinity actually increases conceptually. Okay, so moving on to the heme and porphyrin ring. So a heme is essentially this porphyrin ring with the central iron atom inside of it. And keep in mind the charge. This becomes important because of the his residues, which we'll go into, that keeps it locked at Fe2+. So the heme-containing proteins, heme is a prosthetic group and is associated with myoglobin and hemoglobin. And essentially it's like a functional component of the group itself. And the heme contains two free unbonded coordination sites, distal and proximal, to the plane of the porphyrin ring. So essentially, if we're looking at this diagram or this one as well, we have one above and below respectively for the distal and then the proximal. The distal, and I'll explain this much more in depth in just a little bit. A ligand molecule will bind in Fe from the distal side. And this is because, again, right here, as you can see, and I'll show this in a different diagram because I think it's more clear than this one. The proximal is so close to the iron atom that oxygen just cannot fit in here, right? But on this end, the histidine is further removed from this central iron atom, and that it allows oxygen to bind neatly into here. So that's the difference between proximal and distal, is that the distal has more space from the center atom to the amino acid residue, and because of that extra space, it allows the oxygen or carbon dioxide to fit in neatly, whereas the proximal just doesn't have that room in the first place. So again, heme binds with oxygen and carbon monoxide, plus others we don't really worry about. O2 binds at an angle, and that's because, again, oxygen is not linear. As you can see here, we would have the two lone pairs here and two lone pairs here, and it would create a bent angle. And then CO binds linearly because of the linear structure of carbon dioxide. We only have the lone pair here and lone pair. If you want, just review the Vesper theory stuff. There's a pretty nice video from Organic Chemistry Tutor. I won't link it just because it's pretty easy to find. And then there are two his residues in the heme-containing protein for both of them in terms of hemoglobin and myoglobin. For this one, we have the proximal his, which stabilizes the heme structure by coordinating with the central iron atom. Again, it's super close, so it's going to have much more of an effect and much more of an attractive pull to the iron and lock it in place. And again, this is the most important thing that I recommend that you memorize, is the proximal histidine prevents oxidation of iron from Fe2 to Fe3, which would inactivate the heme. If the heme was essentially a +3 charge, it wouldn't work properly. We need it to be +2 so it can grab the lone pairs from the oxygen as well to coordinate with that. If it was +3, it would be too many, and oxygen wouldn't be able to bind as tightly as it would in the +2 state. So that's something to keep in mind. And this is the proximal histidine, and I'll show you the diagram how you can kind of tell the difference, but this is good to memorize. His F8 is the more proximal one. And then for the distal histidine, it facilitates O2 ligand binding to the heme and disfavors CO binding. So the distal his allows for the bent structure to take place, to be more favorable. Remember that carbon monoxide, how it goes straight up and down, it's linear on this previous diagram. Sorry, let me go back. On this one right here, see how it sticks up almost like a needle essentially, and then this one is more like relaxed, bent. It can kind of turn around, like pivot like a ball and socket. I don't want to get too abstract, but essentially because it's not necessarily straight up like the carbon monoxide, this oxygen, it'll bend slightly to the side so that it allows for more favorable binding with this histidine. Whereas if it was just straight up, it wouldn't bind as favorably. There could be steric clashes, whatnot. So again, the distal histidine influences specific binding affinities by creating steric clash with molecules that would linearly bind to the heme, and aka just carbon monoxide. And as a result, this is the little thing, is that myoglobin binds carbon monoxide only 40 times more tightly than O2. However, a free heme, one that isn't bound to these amino acid residues, just this porphyrin ring plus the iron, it binds carbon monoxide 20,000 times tighter than O2. This is a good fact to know, and if they ever explain why is it that the carbon monoxide's affinity is much less in myoglobin rather than the free heme, use this justification of the histidine residues being there, and specifically the distal histidine and also the proximal as well. Moving on to myoglobin. So this is a structure here on the left. On the green, you can see we have the heme, the porphyrin rings, and also the iron, which it's kind of hard to see here. And then the blue is the alpha helices of the protein surrounding it. And then the red is just the oxygen atoms. So again, myoglobin is just a protein consisting of alpha helices and the heme. The alpha helices have our histidine residues, whereas a porphyrin ring is just the porphyrin ring, no histidine residues, none of that. And then the function of myoglobin is oxygen storage, and it's typically found in the muscles. And it binds oxygen extremely tightly and will only release at very low partial pressures. And we'll see the curves, how it relates, but remember, it's the oxygen storage atom. And again, it functions as oxygen reserves for certain tissues. And a little test for yourself, can you identify which His residue between these two is proximal, which one is distal in the diagram to the left? Again, I was mentioning a little bit before that you can kind of tell spatially which one is the proximal and distal. And from this diagram right here, I would say it's a safe guess that because of how close these two atoms are, the orange is the iron and this one is the histidine atom. I don't know if it's the nitrogen or whatnot. That's not really important. But because this residue is extremely close to the iron atom, I would bet money that this is the proximal. And this one is just so much more further away than the proximal histidine that I would classify this as the distal. Obviously, you can justify however you need to, because this would be like a short edge to respond on the exam, which one is proximal, which one is distal. But that's the justification, is that just based on spatial orientation. And then moving on to hemoglobin. So right here's this neat structure from Pimal where we have essentially the green are the hemes, and notice how there's 4, not 1. This one only has 1, so just keep that in mind. For this one, we have 4 hemes in a tetramer protein, where we have 4 distinct subunits that are related to each other. And again, it's alpha 2, beta 2, which just means that the 2 reds are similar, homologous, and then the blue are similar as well. And note that this naming convention has nothing to do with the alpha helices and beta sheets. It just labels as structural components. I don't know why they chose to, but they chose to nonetheless. And then there are 4 total subunits, as I mentioned, 2 of which are labeled alpha and 2 of which are labeled beta. And each subunit contains a heme and it can bind up to 4 oxygen molecules. Whereas this one, you only see 1 heme. So as you can guess, it probably only binds 1. So good, something to know, like subtle distinctions. And again, it's structurally homologous to myoglobin. The primary amino acid sequence for the 2 proteins is very different, though key amino acids, those histidines, are preserved. And again, here are some other visualizations of it. As you can see, it's beta 1, beta 2. It has nothing to do with beta sheets or anything like that. It's just the way they designated the individual subunits of this tetramer. And then again, this one, the function is oxygen transport, not oxygen storage. And it's typically found in red blood cells. When we talk about a red blood cell, we think of hemoglobin. What's the person's hemoglobin levels? All that stuff. And yeah, again, they look very similar, right, at these heme groups, but that's because they have those preserved amino acids, those conserved amino acids, the histidines that are able to coordinate that porphyrin to fit neatly into this active site. And then just kind of moving on to more general stuff about the hemoglobin itself, there are 2 states, and this brings into cooperativity, allostery, we'll discuss it all. So there are 2 states of hemoglobin. There's a T state and an R state. And it sounds counterintuitive, you just kind of have to memorize it, but T state is called the tense state. It's short for that, and it's the low affinity state. And essentially it's hemoglobin deoxy just means lack of oxygen, just a little analogy to connect it to something else, deoxyribose, right? Without that 2' hydroxyl group, whereas ribose does have that 2' hydroxyl group. Little analogy, you can look it up, you'll see the similarities there. And then again, proximal His is slightly further away, and I'll show you this in the next slide. It's a beautiful diagram. I think that once I show you, it'll cement in your head. So yeah, proximal His is slightly further away. Heme iron is pulled out of the plane. So instead of being flat, the heme, it's kind of bent. Actually, it's bent this way. So if the oxygen were to bind on top, right? If it's flat, it'll bind easily. But if it's bent, it has a steric clash. And so that's something to keep in mind. Whereas the R state is the high affinity state, and R is short for relaxed. I know that's counterintuitive, but that's what they're named for. And essentially, the proximal His is much closer to iron, which allows that bent plane of the porphyrin ring to kind of flatten out now. It's not really visualized here. I'll show you in the next slide. But the heme is more planar, and the iron lies squarely in the center of the heme, which allows oxygen to now bind neatly onto it. And so again, the mnemonic to remember something is R state retains oxygen. It allows that high affinity. Okay, and so here's the better visualization in my opinion at T versus R. So T is in the blue, R is in the red, and ignore these tubes, ignore this bottom part for now. What I want you to focus on is the iron right here, the circles, and specifically the His residues here. This kind of pentagon shape in the blue and the red. And so in the blue specifically, we're looking at this now, we see that the His is slightly further away from the heme, and the heme appears to be slightly warped. So it's not planar anymore. And the O2 is less likely to bind. Again, you could rationalize it as a sterics because it's not planar. But as you can see here on this diagram, we have this blue histidine, and it's further away from this iron molecule. And we're assuming the iron is like right around here. And because it's further away, it's gonna try to pull the iron, but the porphyrin ring is gonna stay here because of either some favorable interactions from the sides of the proteins. I don't really know. I just know that for the T state, that proximal His is further away, and then in this red one, it's closer to the iron, which allows it to have this planar shape. It's not pulling that much of a distance anymore, if that makes sense. So you can see it's 0.6 angstroms. You don't have to memorize that number. Just pointing this out is that the histidine in the R state is much closer. And so if you want to see more familiar diagrams, slides 18 to 19 on your lecture 4 are going to be your best go-tos. And then talking about allostery, how this relates with hemoglobin. So allostery, in simple terms, is that when one molecule binds to a protein, or the ligand binds to a protein, it changes the chemical affinities, or just the affinities of that protein itself. So what I mean is, remember hemoglobin, how it has these 4 distinct units, right here. It has 1, 2, 3, 4. If one oxygen atom binds to one, it's going to affect the affinity of these 3 as well. So it's subtly hinting at that cooperative binding, but that's what allostery means, is that if an oxygen binds here, it's going to affect the affinity of these 3 by conformationally changing and altering the structure of the protein. So that's something to keep in mind as we go through this. So allosteric protein contains binding sites in addition to the active site. So that's how we're able to see these properties of allostery and how one molecule can affect the binding affinities of the rest. So essentially the way to think about this is consider a heme group experiencing binding to be the active site and treat the other 3 heme subunits as allosteric sites. That's the best way to do it. And remember that myoglobin only has 1 heme, right? And we see that there's these different shapes, not necessarily the KDs or anything, just the different shapes. This one is hyperbolic, this one's sigmoidal. So the sigmoidal is the hemoglobin, right? And myoglobin is the hyperbolic. It's very subtle, the difference. But what I'm trying to show is that for myoglobin, because of there's this one subunit, you don't see the characteristic sigmoidal, which indicates that there's changing affinities within this protein. And it makes sense. Myoglobin only has 1 subunit, therefore it's not going to affect the other subunits because there are none to begin with. Whereas hemoglobin has 4 subunits, but if we have one that binds onto it, it's going to affect the other 3. And then if 2 bind onto it, it's going to affect the other 2 to be more cooperative and to saturate at a much faster rate. So we'll talk more about how this relates in terms of this graph. We'll dive deeper into it. So cooperative binding essentially is just oxygen binding to one subunit induces that conformational change in the hemoglobin, which promotes affinity to the other 3 sites, if that makes sense. And because of this, we see the sigmoidal curve for cooperative binding. So we'll go into this. So why is the curve sigmoidal for a cooperative binding protein, such as hemoglobin? So for sigmoid, we see that there's a start phase close to 0 and a shallow slope. Slope is very close to 0, and that indicates low affinity. We'll think about it. But again, as we go through this graph, you can think of it as first derivatives, right? So right now, the first derivative is close to 0, so it means it's low affinity, right? First derivative, we're saying that it's kind of proportional to the binding affinity. Something just, I don't know if this is true or not. This is just the way I think of it conceptually. But essentially, the initial phase is when we're increasing with progressively steeper slopes. So that first derivative, starting from 0, but we're reaching the maximum here, the maximum, the local maxima, I think that's what it's called. I don't really know. But the slope is going to start increasing. And that means that the affinity is actually increasing itself. Why is that, right? As you can see here, we're starting off with a high affinity, and then it starts to drop off. So we're not even seeing cooperative binding here in the first derivative. That's the way I'm phrasing this. And so, again, note that you don't have to know this in extensive detail. I'm just helping you interpret this cooperative binding so it makes more intuitive sense. So as we reach and go from the start to the initial phase, we're going to reach this maximum point as designated by the line. And this is sort of hinting at the half of the saturation. Again, that theta is equal to half. That's when we start to see that, okay, now the slope is decreasing instead, going towards 0, rather than trying to just increase and increase to infinity, right? So that transition phase is increasing the saturation levels, so that y-axis is still increasing, but the slope, that first derivative, is decreasing, which means that the binding affinity is now lowering. And so at the very end, we see that the graph levels off at near full capacity in a flat slope. And this makes sense because if all the oxygens bind to it, then why would it have higher affinity for more oxygens? That would just keep it locked in place. Remember, the function of hemoglobin is to load itself with oxygen, the 4 if possible, and to deliver it to tissues. So at a certain point, it has to get rid of it at a certain point. So it doesn't make sense for it to continue having that high binding affinity just because how's it going to get rid of it later on? And so the initial phase with increasing slope corresponds to positive cooperativity as more ligand molecules bind to the hemoglobin. And we'll talk about this in terms of Hill coefficients and all that. But I just want to let you know that that first derivative is, you could think of it conceptually, if this helps you, as kind of proportional to the binding affinity. And then as the transition phase with decreasing slope corresponds to greater saturation of binding sites, it results in a slower increase of binding affinity as you go on until you hit max saturation. And that's just because there's fewer binding sites available. And then the plot always increases. So there, you never see a point where it goes back down. When it's offloading, you just go in the reverse direction. But as it's gaining the binding sites, it doesn't go down and then back up. It never offloads anything. So protein bites are always becoming increasingly saturated, if that makes sense. So kind of distinguishing adult hemoglobin, which is what we were just talking about from the start till now, we're now going to talk about fetal hemoglobin. And essentially, fetal hemoglobin doesn't have beta subunits. They have gamma subunits instead. So remember that we had this alpha right here, this alpha 2 beta 2. Instead of alpha 2 beta 2, we now have alpha 2 gamma 2. And so this is important because it's a different isoform of the subunit. Isoform just simply means it's a similar in terms of function, but it has it's similar in function, but it has a slightly different structure to it, protein structure. And it doesn't bind 2,3-BPG, which we'll go into in a little bit. This is just a fact you should know now. You can come back to the slide in a little bit. You can just write a little star saying, come back to this after we finish lecture for conference review. And then for fetal hemoglobin, it has higher affinity for O2 than adult hemoglobin. And why is that? Think about it in the development point of view, right? If the fetus is developing inside of the body, it's not going to be able to breathe air on its own, right? What's going to end up happening is the oxygen that the mom is able to breathe in through her lungs is going to get transported via the umbilical vein, umbilical arteries to the infant. And so the infant needs to grab on as much oxygen because it's developing too. It needs that oxygen to do all the metabolic processes. And because it's developing, it's in an anabolic stage. So it needs that high influx of oxygen. So hemoglobin naturally, structurally, is going to have that higher affinity for the infant rather than the adult because it wants to kind of strip off the oxygens from the adult hemoglobin and keep it for itself. And this just makes sense in a developmental standpoint. So something to keep in note is that fetal hemoglobin is much, much higher affinity for oxygen than the adult one. And it just has to do with development. The baby cannot breathe on its own and inside the womb and all that. So something to keep in mind as well. And this graph illustrates that. Remember again, if the P50, the K, you can just assume this is the KD, and this lines correspond to that. At the 50% saturation, if the KD is less for the fetal than the adult, that means that the binding affinity, again, it's inverse. So if it's less, binding affinity is higher. So this makes sense that the fetal hemoglobin is much, much higher than the adult. And then in terms of the myoglobin, obviously it's going to have the highest affinity because it's a one subunit. It'll just take the one, and it's an oxygen storage, not an oxygen transport. Yeah, and yeah, that's all that you really need to know from this slide. Yeah, and then really keep in mind. You'll get something like this where you get the subunits. It's not arbitrary, but check the concentrations of the x-axis when each in halfway saturated y-axis. So essentially when you reach that 50%, you're going to basically draw the line and then draw straight down, and then whatever the unit is for the x-axis, keep that in mind. So here it'll be 2 millimeters Hg. The other one will be 1927. So keep that in mind. If you have to make approximate estimates, the professor's not going to fault you for it. It's as long as you demonstrate that you understand KD, binding affinities, and all that stuff. Okay, and then here is just a summary of hemoglobin versus myoglobin. You guys can go through this. I put it as an animation specifically so you can do one at a time and just test yourself, but I'll just briefly just go over all this. You can review this later. It just talks about everything that we were talking about. And then also another pop quiz testing you on your amino acids. So based on these structures, can you recognize and give me the 3-letter codes and the 1-letter codes for each one? Okay, so moving on to cooperativity. This will explain that sigmoidal graph much more in depth and how that first derivative is correlated to binding affinity. So cooperativity is when the binding of a ligand to one subunit affects the binding affinity to the other subunits. Again, that's established. You guys know this already by now. So the Hill coefficient is the measure of the degree of cooperativity. So it only applies to ligands that are also an allosteric regulator. What that means is that if we have the Hill coefficient being equal to 1, there's no cooperativity. Hence why we see myoglobin continuously have NH is equal to 1. It is never at 3, it is never at a negative value, it is at 1, demonstrating that there is no cooperativity, and this makes sense. It's a one subunit. The binding of a ligand to one subunit affects the binding affinity of other subunits. Well, if it's only one subunit, there's no other ones for it to change. Does that make sense? And then for hemoglobin specifically, we see that the NH right here, if it's greater than 1, it has a positive cooperativity, right? And then if the NH is less than 1, it is negative cooperativity. So what positive just means is the more molecules bind, the higher the affinity increases, right? And negative cooperativity is the more the molecules bind or dissociate, the more it's going to want to dissociate. So in this instance right here, what we see is that the Hill coefficient is approximately equal to 1 when hemoglobin's in the low affinity state. What letter is that? I'll let you think about it for a moment. It would be the T state, the 10th state. So at the T state, we see that the hemoglobin has low affinity and the Hill coefficient is equal to 1. At a certain partial pressure, we start hitting that one subunit being fulfilled, right? That's where this corresponding thing is equal to 1, right? So we see that we have enough equivalent oxygens to fill up the one binding site. Because that one oxygen binds, we now see the Hill coefficient increasing to 3, this slope, almost like the first derivative. We see that first Hill coefficient increase to 3, which means that it's greater than 1. There's positive cooperativity, right? So because that there's positive cooperativity, that first oxygen binding has prompted the hemoglobin to increase the binding affinity for the 2 and 3 subunits, right? So from the 1 until it reaches to the 2, 3, it'll remain positively cooperative. And that binding affinity is going to be extremely increased. Then after a certain point, a lot of these sites are going to become saturated. And what that means is that we're going to return back to the low affinity state. The high affinity state with no cooperativity. And interpreting the chart, the HB line, which is red here, is a composite of the 3 dashed lines indicating the 3 different states of hemoglobin, where it has low affinity, no cooperativity, low to high affinity, and it's making that transition, but high cooperativity, and then the high affinity, no cooperativity. Hopefully that makes sense. I didn't just say a bunch of words. So moving on to the next one, unpacking the Hill plot a little bit further. The HB plot red is a composite of the 3, and the graph transitions are marked in green circles here. So the first transition is from N1 to NH3, and so there's no cooperativity to positive cooperativity, going from this to here. So we see that the oxygen bind conformation changes and enhances binding sites to other unoccupied binding sites. So the 1 binds, it changes that shape of the protein. The other 3 now say we want oxygen. So something to keep in mind conceptually. And then the second transition is, as we have positive cooperativity, we go to no. And it goes from NH equals to 3 to NH is equal to 1. And the binding sites are saturated now, which lessens the affinity for oxygen binding. It doesn't make sense that once it's saturated, again, the function of hemoglobin is to transport oxygen to the rest of the body. At a certain point, it has to offload it. If the binding affinity remained extremely high for an infinity until it's fully saturated, then how would it ever offload it? So it has to go back to this no cooperativity state. And yeah, again, try to correspond the phases of the sigmoidal binding curve with this one right here. So once you figure that out, you kind of have this concept solidified. And don't worry about the mathematical derivatives of this. Just know that the Hill coefficient, how to identify it on a plot. So essentially, there's a bunch of this stuff here. You don't necessarily need to know other than this, where we know that the NH equal to 1 is going to be just a straight line slope, and it's going to be pretty much a y equals x curve, if that makes sense to you, if you know what that looks like. I mean, if you look at y equals mx plus b, it's just y equals x, slope is equal to 1, and then plus b has a y-intercept. And then if it's not that, then we know that it's positive, cooperative, or negative. And you'll know the negative is because it's going to have a less steep slope. And then in terms of the concerted and sequential models, these are ways that explain cooperative binding within hemoglobin. So concerted is all subunits are in the same state, and there's basically just an all or nothing. And subunits are either all in high affinity or in low affinity. So in this case, we see that we could have the potential route of first ligand binds and nothing changes about the affinity state. Second ligand binds, no change in affinity. Third binding, no change in affinity. Fourth binding, and it's fully saturated, no low affinity. But then all of a sudden it switches to high affinity. We don't know which one is true or false. We don't know. But these are just models to think about this high affinity in relation to ligand binding. And of course, like we could have it just change to high affinity all of a sudden with no ligands binding. And then from there, the ligands begin to bind. And then sequential suggests that the affinity, it's not an all or nothing response. It could be individual subunits, right? And yeah, each subunit oscillates between the high and low states independently, one by one, and the binding of oxygen changes interactions of the subunits, and binding of oxygen shifts equilibrium more towards high affinity. And just like a simple note, like the real explanation lies somewhere in between these two models, and we just don't have a full understanding of it. It could very well be this diagonal portion where one ligand binds, it increases the high binding affinity of that one, and the second one binds, and then so on and so forth. It could be that. We have no idea. But for the exam, just know that the two models exist and what differentiates them. One is all or nothing. The other one is more individualistic and separate, one by one. And now moving on to the Bohr effects, factors affecting hemoglobin binding affinity. We have 4 factors. I'll explain them much more in depth. But the first one is carbon dioxide, pH, 2,3 BPG, DPG, however you want to designate it, and then temperature. And here we have a nice brief summary diagram. I try to remember this because it's at a certain point, it gets really convoluted with the different manipulations, and just understanding that a left shift means increased affinity for O2, and these are out of the 4 factors, this is what needs to happen to each one in order for the affinity for oxygen to increase. And likewise, on the other hand, we have the right shift, which means that there's a decreased affinity and its corresponding manipulations to the 4 factors. And yeah, again, use this helpful figure to help you learn the factors and trends. It's going to be able to ground you in understanding why increasing the pressure of carbon dioxide leads to a decreased affinity in oxygen. So something to keep in mind as we go through this. So moving on to the next slide, we're going on to factors affecting hemoglobin binding affinity, CO2, and pH. So in the big picture, we have higher levels of CO2 and H+ that stabilize the T state of hemoglobin, which reduces its affinity for oxygen. So if things favor the T state of hemoglobin, we know that it has a reduced affinity for oxygen. Again, it has to do with the planar stuff. It's now no longer planar in the T state, and there could be stereo clashes. I have no idea. And then in terms of CO2, it can bind to the amino terminus of hemoglobin residue, and this forms a carbon amino species that favors the low affinity T state structure. Essentially, CO2 binds favorably, that allows it to change the hemoglobin to low affinity state. That's all you have to really know. And so the Bohr effect relates pH and CO2. And essentially, remember this graph, how I mentioned that by increasing the partial pressure of carbon dioxide, we have decreased affinity of O2? Well, that's because of the fact that carbon dioxide will bind to the hemoglobin and promote the T state, but also this can relate to pH. Remember, in the other one, and I'll tell you by memory, that the pH, if it's lower, that means we have a higher concentration of these hydronium ions, right? The H pluses, the protons. Sorry. If we increase the concentration of protons, the reason why it also relates to these two is because remember this bicarbonate buffer system, carbonic acid buffer system? Well, if we increase this concentration within the blood, the hydrogen ion, we see that's going to shift it to the left. And what ends up happening is we end up increasing this concentration of carbonic acid, and it's not going to go back because this is already high as it is, the concentration. So it's going to use that carbonic anhydrase within the blood or within the cells, and it's going to convert it into CO2. And when it converts into CO2, we already know it binds to this amino terminus. So we see how these two relate now to each other, is that when we increase the hydrogen concentration, we push it to the left, it drives up the CO2 interactions, and the CO2, if it's present, it will bind onto the hemoglobin and decrease the affinity for oxygen. And this makes sense. When you're exercising, you're breathing a lot of carbon dioxide, you're breathing it out, you want the hemoglobin to release the blood into your muscles. It makes sense, like in terms of physiologically how it's applicable to our everyday life. And also something to note is that the hydrogen ion itself also binds to the His residues in hemoglobin, which favors that low affinity T state. So not only do you have this shifting towards carbon dioxide, you're also having its own effects once it meets with that hemoglobin within the blood plasma. And this is due to the favorable ionic interactions between the neighboring side chains. If you look at it, it has to do with an aspartate and a histidine residue. So it's pretty interesting to look at. It's in the lecture notes. But yeah, and this is an equation showing that the hydrogen ion is displacing the oxygen as well. And another factor, the other two factors affecting HB binding affinity, BPG, and temperature, the body secretes 2,3-BPG, which is 2,3-bisphosphoglycerate. You'll learn about this in glycolysis. It's not really, you just need to know 2,3-BPG for now. The body secretes this in response to higher altitudes, when we have lower oxygen concentrations in higher altitudes. So it fits in this donut hole. There was a diagram that you guys had, but essentially 2,3-BPG, the hemoglobin has a hole within the middle. As you can see within these 4 subunits, it has this hole in the middle that 2,3-BPG fits into. And that's why we say donut hole. And this is in the T state, right? So it promotes and maintains this T state, resulting in a lower hemoglobin affinity for oxygen. So when it's R state, it kind of it condenses, and that 2,3-BPG can no longer bind. However, when it's in the T state, it locks it in place by staying there, and it can't condense anymore. That's realistically how you can think of it. And then there's temperature as well, which can affect protein stability. As you know, if you increase the temperature, it's going to denature the protein. And so higher temperatures favors the T state of hemoglobin, while lower temperatures are favorable to the R state. And this makes sense. When we're working out, we get really sweaty, we get really hot, our internal temperature increases a lot. Well, when we're exercising and our temperature goes up, we need the oxygen to be delivered to our muscles. So it's almost kind of like an alternative explanation that just makes sense physiologically, that when we work out, we increase the temperature, we need the oxygen, we need hemoglobin to have this low affinity state so oxygen can be delivered to our muscle tissues. And again, higher temperature decreases O2 binding affinity, and lower temperature increases it. So again, if you ever get confused, refer to this graph, memorize it, like this, ground yourself in this graph, and then use these 2 slides to explain it to yourself why each state is favored. And then some study exam tips for factors affecting HB of binding affinity. It can be helpful to think about hemoglobin binding affinity in the context of exercise. Again, I mentioned a bunch of them, but during exercise, you want to unload oxygen from hemoglobin to your tissues. And so when you're think of all these factors as how to decrease O2 binding affinity, to unload it from hemoglobin to your skeletal muscle tissues. And so exercising increases body temperature, CO2 production, acid, and you can think of it as like lactic acid, right? It's just proton concentrations. And these exercise related trends are associated with decreased O2 binding affinity. So you can always ground yourself in physiologically exercise. Blah. You can ground these in exercise, and it'll just make sense. But key thing about exams is be explicit and mention TR state in your exam responses. If a question asks you how to change in one of the 4 factors affecting binding affinity, discuss which form of hemoglobin is favored, so T or R, and connect that to higher lower affinity for O2. Connect the structure of hemoglobin to the binding affinity of hemoglobin and oxygen delivery. So example, don't just write BPG lowers binding affinity for O2. No. Right, BPG promotes the formation of the T state by fitting into the donut hole of hemoglobin, which consequently lowers the binding affinity for O2. So something to keep in mind is that you always want to be more explicit about your rationalizations for biochem exams. You don't want to just put the bare minimum. It's always better, good principle for this class, to put more, just to make sure that you don't lose points on simple things that you knew, but you didn't explicitly write out and you thought it was implicit. Always be explicit about your answers. And then just a little fun example that we had last year that I thought we should just continue to keep is the sickle cell anemia. And it's a point mutation in it. And essentially just how this mutation even occurs, it's a point mutation in a gene that encodes for the beta hemoglobin subunit. It turns it from glutamate to valine. And the question is, what amino acid properties change this mutation? This is for you to figure out. I'll let you pause the video now and just explain what happens to these mutations. But essentially, the glutamate is negatively charged, and the valine is no charge at all. So you're turning it from a negative charge to a neutral charge. So you can think of, potentially what are some of the results? What favorable interactions do you lose that stabilize hemoglobin's shape, right? Well, glutamate's negatively charged. The histidines are positive. It could be that the glutamate coordinates those histidines to have the proper spatial orientation, and that allows it to be coordinated to the iron atom. So you can think of it like that. When you mutate it to valine, it loses that negative electrostatic interactions. You can talk something about like that. And again, sickle cell anemia, when you have this mutation, it leads to the aggregation of hemoglobin. So it creates this misshaped red blood cells that get stuck in blood vessels and cannot efficiently carry oxygen to the tissues. Again, has to do with the hemes, the coordination of the iron and the oxygen. It doesn't work because of this mutation. So that's what sickle cell anemia is. That was relevant to this lecture. But moving on to lecture 5. Oh, man. You stretch. Lecture 5 is enzyme kinetics. This one, it's at first hard to understand because it's a lot of details, but I'm going to give you just the overviews. You can go into the lecture slides and learn the rest of the derivations, all that stuff. You're not going to be given a proof on the exam. You could. I think it's fair game, but I've never seen it on an exam before. That's all I'm saying. I haven't seen that people or the professors would say, write the derivation of the Michaelis-Menten equation from start to finish. Given this, I've never seen that before, and I think that's unfair if they test you on that, because that's not the point of the class, to learn how to apply it. So giving you guys just like the basics of reaction dynamics and how this relates to enzyme kinetics. We know that delta G is a standard free energy, and for our purposes, when we use the apostrophe and the not symbol, it just means that it's at physiological pH. pH is equal to 7. So we know that negative delta G, spontaneous, exergonic. Positive delta G is non-spontaneous, endergonic. So at equilibrium for these, the products will be greater in the spontaneous process, whereas the substrates will be greater in the non-spontaneous. And this makes sense. If it's non-spontaneous, it doesn't want to happen on its own, it's not going to want to go to the products. It's going to want to stay at the substrates. And inversely, if it does want to happen, if we do want to go from substrates to products, like here, this is an exergonic reaction, spontaneous, then it's going to want to do this if it has enough activation energy. And so this is a relation delta G is equal to delta H minus T delta S. That's just how to combine temperature, entropy, and enthalpy. And then, as we were talking about equilibrium, the equilibrium constant at physiological pH is simply just the products divided by substrates. We already talked about association, dissociation, product ligand divided by PL. And so the way to relate the equilibrium constant is we have delta G naught, or standard, whatever, is equal to negative RT natural log of the equilibrium constant. And again, there's this inverse relationship of KQ and delta G. So if KQ is large, that means that delta G is negative, right? And this should make sense. The KQ is the products divided by substrates. If our KQ is a large number, that means the products are this value right here is much greater than the substrate, the bottom, the denominator. And that's a whole number. If it was 1 to 1, it would be 1. And if you increase the top, then it's 2 to 1. Now it's 2. So that's the way you can think about it. And so if we increase the KQ, our products go up. And that means that the delta G is thus going to be negative. Whereas if we have KQ is going to be, like 0.1, right? Because we can never have a negative number for this, because you can't have negative substrates or negative products. But we only go to 0. But anywhere from 0 to 1, so it could be like a decimal, it's going to invert this sign. So I know it's negative here, but if you have any number from 0 to 1 and you take a natural log of it, you're going to basically invert this, and now delta G becomes positive. What do we know about a positive delta G? It's non-spontaneous. That means that at equilibrium, it's going to favor the substrates. So just know that there's this inverse relationship. Play around with it. It should be pretty self-explanatory. There are a bunch of resources online about this. And then transition state. So something to note is that these are short-lived, unstable molecules that are very high energy. You cannot put them in a bottle and just keep them for later at all. That's the best way to think about it. And why is this exactly? It's because when we break stable bonds, order conformation of molecules, destroyed favorable interactions, when we destroy everything that stabilizes the substrate, it's going to want to like go somewhere, either back to the substrate. When it's here, it'll either go back to the substrate, or it's just going to want to go to the products. So it's never going to want to stay at this high energy state. And so there's an equal probability of forming products or going back to the substrate. We don't know exactly what's going to happen. And absolute high of transition state energy determines the rate of the reaction. So the only part of kinetics about this is that if this is really high, it's probably going to be slow. We don't know how much exactly. There's no experimental calculation we can do, but we can see that on the next few slides, like here, for example. This is just comparison. We'll go back. But on this one, we see a huge hump if there's no enzyme here. If it's just a substrate on its own to go to the products, there's a huge activation energy. Like, it is possible for this to go and this reaction to happen spontaneously, but it's going to happen at such a slow rate. Whereas if we introduce an enzyme, it's going to make these favorable interactions that cause the enzyme to form this product, right? So for example, instead of having huge activation energy, we now have just a smaller. And so that, because it's smaller activation energy, we need less overall, and that will increase the rate of the reaction. Because if we're at a stable temperature for both these conditions, one with an enzyme and one without, the one with the enzyme is probably going to happen faster, just because we have that extra energy being able to allocate to different reactions. Hopefully that wasn't too abstract. That's just the way I conceptualize it, but you can replay that again and see what I'm trying to say. Or just ignore it. That was just trying to go too far ahead. But, so absolute, yeah. Relative heights of energy to go from S to the transition state and then P to the transition state determine the equilibrium, the thermodynamics. So for example, right here, we see that this delta G double dagger, I was forgetting it. Delta G double dagger, S to P is going to be from the substrate line to the transition state. So we have that. And then we also have the delta G double dagger, P to S, or this should be P to, reverse direction, is going to go up to the transition state. And you can see that it's a larger gap, right? And so when we take this value and subtract it from this one, or either or, I can't remember, that's not important, we see that there's a difference in this delta G naught. And that's exactly what our standard free energy change at physiological pH is. And why this matters is that it tells us whether this reaction is spontaneous or non-spontaneous, and that will tell us where the equilibrium lies. For example, in this one, we know it's spontaneous. Delta G is negative. Therefore, the equilibrium constant will be positive. What does that mean if an equilibrium constant is positive? It means that the products were favored. So that's something to keep in mind. And now let's just reverse this, right? We were saying this was S and this was P, and we were going in this direction, right? Well, the delta G would now be positive. Your end result would be more positive than the starting delta G. And what this means is that because it's positive, we have now a non-spontaneous process. So our delta G is positive here, and we have a negative. So we're going to take the negative, move it to the side, and KQ is now going to be negative. And if we take the E of a negative number, we just know that it's any number from 0 to 1. And because that it's a number from 0 to 1, this is going to be a higher number. The substrate is going to be a higher number than our products. So we're going to have a reaction at equilibrium that favors the substrates rather than the products. So hopefully that makes sense. Like, that was just a lot of abstract me yapping, but just walk yourself through this diagram in terms of delta G's and specifically the delta G's of the double daggers and also delta G knots, and how that informs you of the thermodynamics of it, whether it's spontaneous exergonic or non-spontaneous endergonic, and then how that relates to the equilibrium constant. And then the role of enzymes, as I mentioned before, the enzymes will just lower the activation energy required to reach the transition state. And this is like an alternative pathway. So this can still occur on its own, just at a much less probable likelihood. Whereas this one will probably occur more frequently just because of that lower activation energy. Again, this increases the rate. It does not affect the equilibrium constant. Again, the free energy at the substrate and the free energy at the products remain the same. So delta G dagger decreases, right? So in this instance, we see that the transition state is going to be much less energy than the uncatalyzed version. And then the delta G of the reaction remains unchanged. Again, that delta G is going to be the difference between here, this level at P, and then to here, to the S. So it's going to be this little gap right here. Hopefully that's right. And then why is it about to have a very stable enzyme substrate complex? I'll let you think about it for a bit. You can pause the video. Just think about why this is bad. But the answer is because if the enzyme substrate complex, this thing right here, is stable, then it's not going to want to go to the EP because it just likes to stay at this energy. It's extremely favorable. So the enzyme, it itself, when it forms this complex, it cannot be extremely stable or else it won't proceed to the product's reaction, you know? So that's something to keep in mind as well, is it can't be too stable because it won't proceed forward the reaction. It'll just stay halted at this ES complex. Okay. Now moving on to the Michaelis-Menten stuff, because I yapped a lot about the thermo, but I think the thermodynamics really sets yourself up to understand a lot of these concepts. So VO and steady state, right? So V naught, or VO, sorry, is the initial velocity, which just means the rate of the reaction at a certain point in time, right? It could be at the start, it could be at the end. It's just proportional to the concentration of ES, essentially. And the assumptions are for Michaelis-Menten, and it's listed here on this graph on the top right here with K1, K2, K-1, K-2, all that stuff. And it's in black or it's grayed out. Only an insignificant amount of product has formed. What this means is that this is such a low concentration that our K3 we don't consider, our K-3 we don't consider, and our K-2 we don't consider reverse because we're saying that there's nothing there, right? Whereas ES, this is going to be the main dominant form, and it's going to form this EP. And that we're talking about this part of the graph right here, where we have our total enzyme concentration, stuff that's, and this is the free enzyme concentration. This is the stuff that the enzyme that is not occupied by the ligand, the substrate. And so it's going to decrease as the substrate concentration increases, right? And this is what the y-axis is, that substrate concentration, and T is the time. As we increase substrate, we see that the unbound is going to form this EX complex, and that's why we see this red line increase. Whereas the blue line, it's going to form this low amount of free enzyme that can bind to the substrate when it needs to, right? So we see that the derivative of this EX with respect to time is the slope of it essentially is equal to 0, and that just means that VO is proportional to ES. So that initial velocity, that taking the slope, right, it's 0, so it just means that it's proportional to ES, and that the V naught is going to, or V0, sorry, is going to be equal to the K2 multiplied by the ES. And so that's just something that you have to remain. And this is called steady state approximations, where the rate of ES formation is equal to the rate of breakdown of ES. So this ES right here is stagnant, and this E is stagnant. And then eventually, once the substrate concentrations diminish, the ES is going to lower and the free enzyme concentration is going to increase. That is outside of steady state. And remember, steady state approximation does not mean it is static. It means that it is a dynamic process where the ES will lose it and then the E will take it, that whatever's free. So it's always like an interchanging thing, but the net is stagnating, if that makes sense. So if we look at it at like a microscopic level, we'll see this exchanges of substrate to the free enzyme from the enzyme substrate complex. We'll see those exchanges at a micro level, but on the macro, you don't really see it. And then Michaelis-Menten kinetics, memorize this equation, please. It'll be on the equation sheet that you guys will get, but please take the time to really understand this, what Vmax means, what Km means, what the substrate concentration does, what would happen to this equation because it's equal to V0. What would happen to V0 if we manipulated one of these values? So please, just walk through this. So Vmax is the maximum velocity of a reaction that occurs with a given enzyme concentration. What this means is that what is a velocity if we have like a flask, right? And we put a bunch of enzymes inside of it. Let's say we put 1,000 with the flask. We're trying to say what is the rate if all of the enzymes were occupied with the substrate and we're converting it to product. That's what Vmax means. What V naught means in relation, or V0, sorry, I keep saying not. What V0 means is that, let's say we have the flask of 1,000, but we only put enough ligands to saturate 50 of them. Well, the V0 is when the rate at which those substrates that are occupied only by 50 of those enzymes, that's, no, I can say this better. So V0, what it means is that, let's say we have that flask again of 1,000 enzymes, right? And it's free. Let's say we put enough substrate that only 500 of those were occupied, right? So we're now at half. So V0 is just what is the rate of the reactions inside the flask that are converting the substrate to product when we only have enough to saturate only 500 of the enzymes rather than the 1,000. So it's only at half. And that's exactly what this is what I was alluding to here, is that when we have half of the Vmax, so Vmax is up here, we take half of that value, and then we draw a line to the curve that we have, this Michaelis-Menten curve, and then we go straight down, that gives us the Km. If you plug in the values, like let's say you have 1,000, right? You put 1,000 here, and then you put 500 here. You'll find out that the Km is going to be equal to the substrate concentration, at which gives you half this Vmax. So Vmax is the maximum velocity of a reaction that can occur with a given enzyme concentration. So when is Vmax reached? It's when all the enzyme active sites are saturated with substrate. They're full. In the graph, when the curve on the right plateaus, that's when you have reached Vmax. As you can see, it's still increasing, so it doesn't look like it's plateauing yet. It's going to increase up to the Vmax. So you kind of have to approximate. And then Km is the Michaelis-Menten constant, and it's the apparent dissociation constant, which is a measure of substrate affinity for enzyme. And it's about equal to Kd when K2 is much, much, much less than K negative 1. If we go back to this graph, when K2 is much, much less than K negative 1, what this and remember the P plus L is PS and dissociation was the reverse. Well, this is what we're saying, is that K2 is so minuscule that K negative 1 is the dominating force of ES, right? And this makes sense. We're saying that this is a very we're at this point we're saying that there are some EP forming, that this K2 is now decreasing because of the K negative 2. And so we're seeing now some of the ES driving more towards K negative 1. And so the apparent the Km is also known as the apparent dissociation constant. And the one way to calculate it is taking that K2 plus K negative 1, K2, either the ES goes to the product or the ES goes to the separated version, and you're dividing it by K1 to go to the ES. Again, remember, we're assuming Michaelis-Menten assumptions, which just means that we don't have the K negative 2. This is insignificant. So these are the three that we take into account when we're calculating the Km. And again, if you want to look at it from the graph itself, we know that the substrate concentration at 1/2 Vmax is going to be equal to our Km. So again, take your Vmax, go to half, and then from there you're going to draw a line straight to your curve, and then go straight down. That'll give you the substrate concentration at which half of the enzyme sites are occupied, and you are now at half of your Vmax. That's what the Km means. And again, thinking back to the association or dissociation constants, that it's inversely related. So if you have, for this case, if you have a low Km, you actually have a high binding affinity. Whereas if you have a high Km, then you have a low binding affinity. You can think of the Km as a Kd, but do know that there's a slightly different. And then, what's it called? How could we raise the Vmax? We know that increasing the concentration of the enzyme will allow us to have more enzymes. So for example, if we have a flask of 1,000, right? Going back to that example, and then we have another flask of 2,000, which one do you think would have a higher Vmax? It would probably be the 2,000 because there's more active sites. There's more substrate. You can increase the substrate concentration, and the velocity of the reactions that are occurring in the flask increases because of those extra enzymes. That's one way to think about it. I know the difference between Km and Kd. Again, I just mentioned that they were subtly different, but I'll explain them here. So Kd is the ligand concentration at which half the binding sites are occupied, whereas Km is the substrate concentration at which a reaction precedes half of its Vmax. Both indicate concentrations at which a desired function for a protein enzyme is half capacity, but in different contexts. Protein binding has to do with the binding sites, whereas Km talks about the Vmax, the reaction rate. So those are subtle distinctions. Now talking more about turnover number and catalytic efficiency. For turnover, we're assuming the number of molecules or moles of substrate converted into product per mole of enzyme at Vmax. What that means is if we have the flask of 1,000 enzymes, right? We're saying a turnover number is, let's assume, this is hypothetical, follow along with me. We have that flask of 1,000 enzymes, right? And let's say that we have all the sites occupied, but they haven't been converted to product yet, right? We're saying we're pausing the button and it forms that ES complex, right? Then what we're saying is the turnover number is that transition from the ES to the EP. So turnover number just relates to this K2, right? So as you can see here, I know this is a different equation for showing this, but we're saying that the turnover number is essentially the rate at which we convert the substrate into product. And again, it will always be bound to the enzyme still. It is not the dissociation of the product from the enzyme from this complex. It's not the dissociation, this part. It's not K3. It is just the K2. That's what turnover number means. And so, in other words, after the enzyme is bound, how fast it turns the substrate into product from ES to EP. And so this is an equation to calculate it in case you don't have that K2. You can take the Vmax divided by the total enzyme concentration, and you can manipulate it around. So if you ever are given the turnover number, Vmax, and they say calculate the total enzyme concentration, you can do that, and vice versa. If you have two of the variables and missing one, you can calculate it. And then catalytic efficiency, this is where it comes in handy because it takes into account the binding affinity and how fast it converts the substrate to the product. So let's say we have hypothetical. We have an enzyme that has a really fast turnover rate. It is so fast that converts the substrate into product. It does it really fast. But let's say it has a terrible binding affinity. It binds once every 1,000 molar of the substrate concentration. So even though it's really fast at converting that substrate to product, because it has that low binding affinity, the catalytic efficiency is extremely low, the ratio. Yeah, because high binding affinity, low Km. So it's used to compare efficiency of enzymes. So we can never say based on just the turnover number or just based on the binding affinity which enzyme is better than the other. We could use it to compare it with that metric itself, but we can never say which one is how efficient that enzyme is without these two numbers. And again, a higher V0, this is an equation that you also should have memorized. And a higher V0 means that a higher catalytic efficiency holding other variables constant. So just something to note. And then also the most efficient just means catalytic perfection, and it's the rate. Catalytic perfection only means that the rate is limited by diffusion. That is something to note. And so again, another situation is that you have extremely high binding affinity, but the reaction happens so slow that it's perhaps not the most efficient enzyme. It's kind of getting bottlenecked at that transition from ES to EP. So there are these two separate conditions that you can have an unideal enzyme despite it having a low Km or having a high Kcat. Just something to consider. So if you're ever comparing enzymes based on efficiency, use this, and you're comparing the turnover number, how fast it turns it from the ES to the EP, and also this binding affinity as well. So differentiating Kcat and Vmax. So this is the subtle distinction between the two because I'm sure that you guys have questions about it. So Kcat is essentially the number of substrate molecules converted to product per enzyme active site. And in simple terms, this is more of a reflection of the intrinsic ability of the enzyme to catalyze a reaction. And so think of it as, let's say we only have one, or say we have 10, right? Enzymes. We don't care about it that it has 10 so long as we're looking at the single enzyme itself and we're seeing how fast it converts that substrate into product. That's all we're really focused on with Kcat, with the turnover number. And then the Vmax, it's the maximum rate of an enzymatic reaction under conditions where enzyme is fully saturated with substrate. So in lay terms, at Vmax, assume all enzyme active sites are filled with substrate. They are saturated. And so the enzyme is functioning at max capacity. Does that make sense? Like the enzymatic solution. And so here's an example problem that you guys could be tested on in an exam. An enzyme catalyzes reaction of S into product. Honestly, I'll let you guys just run through this on your own time, but all the answers are listed in the footnotes within this, but that's something to look at. And then moving on to Lineweaver-Burke plots. So on top of that Michaelis-Menten equation that you memorize, the V0 is equal to Vmax multiplied by S divided by Km plus substrate concentration. You're going to also have to memorize this linear equation. You could learn the derivations, but I really recommend to memorize this as well. And then also something to memorize are these intercepts. And please note this negative on the Km, and also know that they're 1/overs. And same with the Vmax as well. Memorize the y-intercept, the x-intercept, this, and even just the x and y axis. I know that's not a very satisfactory answer, and the slope as well. It's not a satisfactory answer, but it is the truth. Like, this is something that you should know because it'll ground your understanding of a new graph if you're given one, right? So again, this is essentially just the regular Michaelis-Menten equation made it difficult to measure. Oh, okay. So there's a reason why we have a Lineweaver-Burke plot in the first place. So the regular equation that you guys saw made it extremely difficult to measure the Km because you don't know exactly when you're reaching the Vmax. And especially if you consider multiple enzymes, how are you supposed to determine it? It gets convoluted and complicated really fast. So Km is easier to be determined using a double reciprocal plot, meaning that we take the 1/over on both sides. And it's used to get the Km and Vmax in experiments with lower substrate concentrations. And you should know x-intercept, y-intercept, slope, And please be careful when you're converting these. When they say what's the km of this Lineweaver-Burke plot, do not leave it as the -1 over km, meaning that don't just take whatever the value says here. Like if it says like -0.1, that is not your km. You have to take the -1 over 0.1 and then do the calculation, and then that'll be your km. So again, the intercepts are reciprocals, but not actual km Vmax values. Take one extra step of arithmetic to calculate these values from the plot. And so some trends. These are some things that I just had to end up putting into my head because I was cramming last minute. But for slope, if you have the increased slope, if it goes upwards, right? If you're taking that for derivative, whatever, increased slope, that means it's a decreased catalytic efficiency. If you have a decreased slope, increased catalytic efficiency. So if you can see here, remember catalytic efficiency was kcat divided by km. Well, in this instance, the slope is km divided by Vmax. And we know that the Vmax is directly proportional to the kcat. So if we're flipping you can think of this as km divided by kcat. Realistically, like if you go back to this equation, this is what I'm reflecting on. This right here, they're basically directly proportional. If you increase the Vmax, you increase the kcat. Yeah. And so from here, if we have km divided by kcat, then we know it's an inverse. So if we increase that ratio, if the slope increases, we know that it's technically increasing the catalytic efficiency because it's inverted. They're inversely related. So keep in mind. And then, so for the x-axis, we have -1 over km, and just some simple things. If you shift it to the right, you're going to increase the km, and if you shift it to the left, you're going to have decreased km, which, again, remember, km is inversely related to the binding affinity. So your increased km is a lower binding affinity. Your decreased km is a higher affinity. And then for the y-axis, we have 1 over Vmax. And if it's shifted up, we have a lower Vmax. And if it's shifted down, we have a higher Vmax. So something to keep in mind. And understanding these trends will make the graph interpretation easier, especially if they give a graph on the exam. But try to rationalize mathematically why a shifting up in the y-axis equates to a lower Vmax, right? So something to keep in mind as you do this whole thing. And then moving on to enzyme inhibition, is inhibition and inactivation are not the same thing. So inhibition is reversible, whereas inactivation is irreversible. So they're both negative regulators because they're in something. You know what I mean? Like inhibition, inactivation. So inhibitor can unbind from an enzyme, so it can bind and unbind. So there's this reversible component. And then inactivators will stay forever bound, and they're also known as like suicide inhibitors from previous years. And so kind of moving on, I made these graphs to the right detailing competitive inhibition, the Michaelis-Menten plot, the Lineweaver-Burke plot, and also just conceptually what it looks like. So the ligand, for competitive inhibition, the ligand and inhibitor are competing for the active site. So on this diagram right here, we have this inhibitor that's binding on the active site, and the substrate can't bind anymore. And so the Vmax is unaffected. Why? You can figure that out on your own. No, kidding. The reason why the Vmax is unaffected is because when the inhibitor binds, it's also reversible too, right? It can unbind. So the substrate can then therefore take its place, and you can have you can have enzymes that are fully saturated with the substrate because they stay bound and with maybe perhaps a little bit of a higher affinity than the inhibitor. And that results in basically, even though the inhibitor is present in the solution, eventually the substrate outcompetes it and it'll form that saturated levels. It just takes longer, higher enzyme or substrate concentration to do so. And for the Km, it increases by a factor of concentration of inhibitor, alpha. They usually will give this number. Don't worry about the alpha stuff. But active sites are taken by inhibitors. And tips on remembering, focus on the slope getting steeper anchored to the y-intercept. So in this instance, remember catalytic efficiency, right? If we introduce a inhibitor, the catalytic efficiency is probably going to get worse just because it's an inhibitor. And we know that the Vmax doesn't change, so the y-intercept has to be the same, right? So it's going to intersect at the same point on the y-axis. And we see that the slope is greater, and because the slope is greater, we see that the Km moves rightward. What do we know about a rightward shift? It means that it has a... what's it called? I don't even remember. See? I get this mixed up all the time. But no, a shiftward right is going to be a... So we know that a shiftward right is going to be an increase in Km, right? And that means decrease in binding affinity. So this makes sense, right? If we look at this Michaelis-Menten plot here, we have the black line is the just regular no inhibitor, and then the red one is the inhibitor present, and it corresponds to this top one. So we see here that the Km has a lower substrate concentration in order to reach 50%. With the inhibitor, it takes a greater concentration. So we know that the Km increases with the inhibitor, decreased binding affinity. So try to rationalize the two, see both, and then if If the slope gets steeper, the x-intercept must change because we're keeping that y-intercept stagnant, or the same. Now moving on to uncompetitive inhibition, we see that the inhibitor binds only to the ES complex. So it stops the ES from forming the EP or distorts the active site. An inhibitor only binds to the allosteric sites after the substrate binds to the active site. So in this diagram here, we see that the substrate binds, there's allosteric sites nearby, and you could think of it as the substrate binds, and then the inhibitor is like, oh, bet, I'm going to bind now. It's not, the inhibitor will not bind on its own separately. It needs that, think of it as like the substrate and the inhibitor interacting as well to also lock in the inhibitor to bind. Does that make sense? They form like favorable interactions here and also within the enzyme's allosteric sites, and that allows the inhibitor to like stay bound. And so it's almost like you need that substrate to be bound as a prerequisite. So as the Vmax decreases, the Km is the Vmax decreases within this. As you can tell from this Lineweaver-Burke plot, we see that the y-intercept goes up, and remember it's 1 over, so even though it may go up, you have to take 1 over that, and it actually means it decreases the Vmax when you calculate it. And then Km is reduced. Why? Think of it as, for example, this is something to note right here. You can see that on the red line the Km is much less than the black line Km with no inhibitor. The reason why the Km is lower is because you can think of it as the inhibitor locking it in this place, right? And it's like artificially increasing the affinity of the enzyme for the free substrate. So this right here, the separated, like think of this as like a concentration, these two species separated. This is going to decrease and this is going to increase, this ES complex, even though it's not going to form that product and dissociate. There's no dissociation. It just stays locked in its place. That's why we see Vmax go down. It stays locked in place. Remember in competitive, I told you that the inhibitor can come off. It's reversible. I mean, the same thing too. The inhibitor here is reversible too, but it stays locked in. It likes to stay in this EX complex. But here, like I was saying before, it likes to come off, and then the substrate can come back in and take over and then have a higher binding affinity. In a similar way, uncompetitive inhibition, what happens is substrate binds, inhibitor stays bound, and it almost stays like locked in its place. It can unbind, of course, but more often than not, you're going to see it stay locked in its place with the substrate inside, and it's going to lower that Vmax because you're not forming the products at the end of the day. And then, for the Lineweaver-Burke plot, something to note as well. Or sorry, for the Km, jumping the gun. The reason why Km decreases is because, again, you're decreasing the concentration of this E plus S, and you're forming just the ES with the inhibitor as well. And so since this concentration is decreased and it's almost like being locked into this, there's a higher attraction for this ES complex because of the inhibitor, you're going to lower that Km because you need less of the substrate because you have that inhibitor present to almost like push it equilibrium-wise towards this direction, right? Anyways, something to note as well with this Lineweaver-Burke plot, the slope doesn't change. I mean, the catalytic efficiency stays the same. So just something to like have memorization wise, like slope, the x-intercepts, y-intercepts, how uncompetitive inhibitors behave versus no competitive inhibitors. Have that memorized, and it'll help you a lot for this exam. And then we have non-competitive inhibition, where a inhibitor, and this is a type of mixed inhibition, where an inhibitor can bind to E or S with equal affinity for each, right? So it can either bind, the inhibitor can bind to the enzyme by itself, or the inhibitor can bind to when the ES complex forms. And so Vmax is always lowered for these scenarios, but the Km is unchanged. Why? Because equal affinity to E or S means that the alpha and the alpha prime are equal to each other, and they cancel out. And this previous one, I didn't mention it before, but this is alpha prime, and the other one before that was alpha. And then, yeah, again, this is type mixed inhibition. This is what the curves look like. As you can tell, the slope does change. The x-intercept stays the same as expected because Km doesn't change, but the Vmaxes change a lot. And it's usually that they lower with the inhibitor present. So just memorize this, but conceptually like understand that it's equal affinity to either or. And then lastly is the mixed inhibitor. So I mentioned before that the non-competitive is a special subtype. That's when there's equal affinity to E or S. But mixed inhibitors can bind to E or S with different affinities for each. Meaning they could bind favorably to just the enzyme by itself, more favorably, or they can bind more favorably to the enzyme with the substrate right here. So it can vary. So Vmax is always lowered for all of these. Km can increase or decrease depending on if the inhibitor binds to either the free enzyme or the enzyme substrate with greater affinity. And that's determined by the ratio, which should be given to you for that. And uncompetitive, it's uncompetitive if it preferentially binds to ES, and then competitive if it binds to free enzyme. To free enzyme. these are like some ways you could think about it extra, but you don't need to like go too much into it. And then think the slope changes without an anchor to a y-intercept, right? So tip, if you know the characteristics of graphs of the other types of inhibition, then it won't be so difficult to figure out on the exam if you have a mixed inhibitor. And that's realistically all you really need, because mixed inhibitors are wonky. It's really weird to work with them. So I would say that, just memorize the other three in this: competitive, uncompetitive, non-competitive, and then you'll know exactly when it's a mixed inhibitor. And here's a helpful picture of all the types, the three at least that I told you to memorize. Here's the competitive, Vmax stays the same, Km decreases, slope decreases. Slopes, and then there's uncompetitive inhibition, slope stays the same, Vmax changes, Km changes, and then non-competitive, where slope changes, Km stays the same, Vmax changes. You can note these subtle, what holds the same, what holds constant, what changes. And then here's some overall trends dealing with the alpha stuff, how you can calculate a parent Km or the parent Vmax given the alpha values if applicable. So definitely like memorize how you can influence the Km Vmax if you were given alpha prime values. And this is elucidated most in the Achieve homeworks. I remember when I took this course and TA'd it last year. And then moving on to the last couple of slides, there is the bi-substrate reaction, the ternary complex. Oh man. Moving on to the next few slides, there is the bi-substrate reaction, which is a ternary complex. So the complex with three different molecules bound together at the same time, and essentially two substrates bound to the same enzyme in either specific or random order. We don't know. This right here is a random order. Their specific could be like E has to bind to S1, and then S2 can bind to this to form it. The E will not bind onto the S2 alone. That's what specific means. But it does, the hallmark of a ternary complex is that it forms this three part complex where and essentially like you need the three to form in order for the products to be formed. You can't just have ES1 then go to EP1 and then it'll just bind to the S2 and do that separately. It can't do that. It has to bind all three in order for the reaction to proceed forward. And then in terms of this graph, right? So when we're increasing S2, it raises the Vmax and decreases the Km for S1. Why is this? Well, and also like the slope decreases. So let's talk about with the slope first, actually. So we know that from the trends, that slide, that if you decrease the slope, the catalytic efficiency increases, right? So this makes sense though, because if we have this enzyme, right, and we increase the first substrate, and we have a fully saturated ES1. Well, if we have such a small concentration of ES2, then this ternary complex will be very hard to form. So the catalytic efficiency is almost like minuscule, right? It's low. But if we start increasing this S2, it's going to happen more rapidly. And as a result, we're going to have this alternative pathway start to dominate and start to take over this ES2 pathway. That's why we see this catalytic efficiency increase, the slope decreasing each step of the way as we increase S2. Again, note the axes. This is about the x-axis is in relation to the first substrate concentration, and then the y-axis is your V0 or Vmax. And again, it raises the Vmax, so Vmax goes down, 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 and we know that it's inversely related if you go down the y-axis to the Vmax. And it decreases Km, meaning that it moves rightward, which means that your affinity increases. Or sorry, Km moves leftward, and the x-axis moves leftward and the Km decreases, which means your affinity increases. Again, it has to do with this alternative pathway. You can go either ES1, which is usually the dominant when this is low concentration, this S2. But once S2 increases, we see that now it has this alternative pathway, which increases its catalytic efficiency. And then moving on to the ping pong reaction, this I was alluding to where E plus S1 happens first, and then we get the first product, and then S2 happens. And then it could also be just like E plus S2, and then it's ES1. It could be either way. But the point is that you have like the first part of the reaction happen first, and then that changes the conformation of the enzyme so that the second part can occur. So it's almost like it's stepwise. You need first one to happen, and then the second one can react and actually do it. So again, so there's no formation of the ternary complex. So you'll see that the slope stays the same. Catalytic efficiency stays the same. This makes sense. When we increase S2, we raise the Vmax and Km for S1. How is it possible that this thing could raise the Vmax? Well, if we increase S2, then when this first reaction happens, we're left with this altered enzyme state, right? And if this was a low concentration, then it would just be stuck at the altered enzyme concentration. And remember, the starting reaction is not this, it's not the E apostrophe at all. It's just E. So we need this to convert back to E, which is, we need the second substrate in order to do so. So if we increase this concentration, then we have more formation of E, and therefore it raises the Vmax because now we have more of this free enzyme available at the end of the reaction, just because we increase one of the intermediates of it, if that makes sense, of the net overall reaction. And then in terms of Km, it raises. So there's a reason why S2, when we add more of it and we increase that concentration, it increases the Km for S1. There's a reason why. And that's because, remember, Km has to do with number of occupied sites divided by, and this, again, it's related to theta from the beginning of lectures. And it relates to number of occupied sites divided by number of total sites in general. And so note that when we're increasing S2, we're increasing the total number of sites occupied. We're saying that E naught is still an occupied site because it's not E. Does that make sense? And so what we're saying is when we add this, we're increasing the availability of this free enzyme. And when we increase that, our theta becomes lower. And because our theta is lower, it means that we need more substrate to increase it. And in a similar way with Km, we know that by increasing the amount of free enzyme, we have to add more substrate in order to reach 50% saturation again. So even though we're not necessarily increasing S1 right away, what we're doing in this graph is manipulating S2. It gives this effect of increasing Km because we're keeping S1 stagnant, but the number of free sites is increasing so much. And that gives it like a pseudo increased Km, lower binding affinity. And yeah, that's about it. Questions, anything like that, just feel free to reach out via my email from the first video. But man, it was a lot, and I felt like I could have explained some things better in my opinion. But hopefully you guys found at least alternate explanations, clarifications, and whatnot. But yeah, I'm hoping that this video was helpful. I'll catch y'all later on the next one.